I'm Daniel Levine, and this is the Bio Report. In most cases, the immune system detects and destroys cells that could become cancerous every day. For cancer to ravage the body, it needs to evade the immune system by shutting off its response. SIRPant Immunotherapeutics is seeking to address cancer by activating and modifying a patient's own macrophages to awaken the immune system to cancer. The activated macrophages are able to recognize and directly attack cancer cells throughout the body and stimulate other parts of the immune system to sustain a prolonged attack. We spoke to Robert Towarnicki, CEO of SIRPant, about the company's immunotherapies, the advantages they offer over other approaches, and why its platform can be applied to other diseases involving immune dysfunction beyond cancer. Robert, thanks for joining us.
Thank you for having me.
We're going to talk about cancer, macrophages, and SIRPant Immunotherapies' efforts to develop a class of macrophage cell immunotherapies. Let's start with macrophages, though. What are they, and what role do they play in the immune system?
Okay, well, let me try to answer that as simply as I can. Macrophages are a white blood cell that plays multiple roles in the body. They're one of the body's first lines of defense when a foreign protein or object is detected. The name is actually derived from the Greek language. Macrophage means large eaters. So they're a very large white cell in our body relative to other white cell types. And their primary initiating focus is that of phagocytosis, or eating of foreign objects, dying cells, and the like. So when a foreign protein presents, that could be a bacteria, or in our case, we're looking for foreign proteins or recognized foreign proteins as cancer. When the macrophage detects those, they will phagocytize or eat that cell or bacteria, whatever that protein-presenting organism is. And they will, obviously, that kills that foreign invader, but it also elicits other responses from the macrophage. A macrophage is kind of like the conductor of an orchestra, if you will. So once it eats the foreign protein, it then initiates a inflammatory response where it did so. That is a call to arms, if you will, calling in other white blood cells from the circulating system, from the bloodstream, to, in a sense, reinforce their efforts. Then the macrophage turns and has another function. It communicates with these recruited white cells, and they could be T cells or B cells, NK cells, a variety of different cells. It communicates the newly found information from eating that foreign protein. And it essentially programs those cells to seek out and destroy other examples of that foreign protein. So in the case of T cells, we've all heard of CAR T, which is a autologous or a self cell that has been harvested and modified to be reintroduced to the body and kill cancers. That's one of the many white cells that gets its education from that macrophage. When the macrophage has accomplished its objectives, it will then die. The white cells continue and expand and go out and do their job. And at the end of, if they're successful, other macrophages will then elicit a wound healing response. So it's quite a talented cell, has many functions, and is key to the entire immune system, and then ultimately leads to the wound healing aspects of healing after the invader is eliminated.
What happens in cancer that enables it to evade the immune system?
Well, that is a very complicated process, and it's one that takes quite a bit of time to develop within the body. The cancer cells acquire genetic mutations. Those mutations add certain aspects to the cells that allow it to survive and expand, generally in an uncontrolled manner, and importantly, evade recognition by our immune system, evade the detection by the macrophage. And much like natural selection in evolution, within our own body, this is an evolving process where the selection for the fittest, those most resistant to being detected and eliminated, are the ones that expand and take over, ultimately winning out and metastasizing, traveling out into other organs, and ultimately leading to death. But it does take a period of time and an accumulation of genetic defects. Hence, things that generally drive genetic defects can be a cause of cancer. For example, we are advised to avoid sunlight because the radiation does in fact create genetic defects and can lead to skin cancers, for example. Smoking, of course, leads to lung cancer. So there are many things that can enhance the ability of a cell to mutate. And when that occurs, we end up with a cancer that can avoid the detection and elimination by our immune system.
We've seen many immunotherapeutic approaches involving different types of immune cells. What's the case for focusing on macrophages?
Well, as I mentioned just a few minutes ago, macrophages are much like the conductor of an orchestra. They bring all of the various immune components to play, and that results in beautiful music. For example, let's take CAR T as a therapy. It's very well known right now. Has been very effective in the treatment of lymphomas. But I would argue that CAR T is much like one instrument, one musician, and that we need more than that to play a symphony. And so current T cell therapies, they have real benefit in lymphoma. They've shown very disappointing results in solid tumors, and we could talk about that in a few moments. But they do not have the ability to eliminate cancer in and of itself, because it's only one approach or one attack on the cancer. T cells have also been very ineffective in solid tumors and in the treatment of solid tumors. And there's a variety of reasons for that as well. Macrophages, on the other hand, can elicit and solicit T cell responses, and the macrophages can educate the T cells to recognize all of the neoantigens presented by a cancer. But it also can educate a B cell to elicit an antibody response against all of the neoantigens, again, presented by the cancer. And the macrophage within the body is the only way you can accomplish this. This cannot be done outside of the body in a laboratory in such a broad application of all of the immune components. And for that reason, we think macrophages are the superior approach to immune response, because it truly employs all of the tools that we have in our immune system.
Is the expectation that your therapy would be developed as monotherapies, or would they be used in combination with other immunotherapies or cancer treatments?
Well, in our preclinical testing in animal models, we have demonstrated that it is an effective therapy as a monotherapy. But we have also shown that it is enhanced in the presence of other adjuvant types of therapies, so that at a given dose, we can clear cancer in the mouse, for example. But if we want to, we can also clear that cancer in a mouse by giving a lower dose of our product, along with an adjuvant such as low-dose focal radiation of an injected tumor, or the employing of the checkpoint blockades, some of the newer drugs such as Keytruda, that help to disable the avoidance systems of the cancer and make our macrophage even more potent. So it can work as a monotherapy. We're currently testing it as a monotherapy with the intent to move it into combination trials, both with radiation therapy and with checkpoint blockade.
We've seen success with CAR T therapies in hematological cancers. Solid tumors have been more challenging. Is there reason to think that a macrophage therapy could be successful in solid tumors?
Yeah, that's actually the most exciting aspect of this technology. When the macrophage, this is theoretical now. This is what we believe in terms of what we're seeing in the animals and what we hope to demonstrate in humans as we speak. The macrophage, I mentioned, elicits antibodies. And those antibodies are targeting against all of the neoantigens, new antigens, foreign antigens presented by the cancer. This in comparison to, example, a monoclonal antibody being given to cancer patients where they're only targeting a single cancer-associated antigen, which allows for those cancer cells, again, to be selectively eliminated, but in the reproduction of the cancer, they can escape mutate, and therefore there tends to be recurrence. We're attacking multiple antibodies or antigens at the same time through multiple polyclonal antibodies. Those antibodies circulate freely in the blood system and go out and find and attack the metastases, the distal tumors. And the antibodies weaken the membranes of the cells and make them more susceptible to T cell penetration. And that's where CAR T has failed in solid tumors. It is unable to penetrate the capsule of the tumors and get inside and do their job and kill the cancer. So we believe the combination of the elicited antibodies in combination with T cells targeting all neoantigens will show a benefit that exceeds anything else being done today in cell therapy. We've certainly seen it in the animals. And as I said, as we speak, we're recruiting patients now in lymphoma studies, and we'll look to recruit patients into solid tumor studies in the near future.
SIRPant has developed platform technology it calls PhagoAct to create its therapies. This is a non-genetic method to activate a person's own macrophages to recognize and attack cancer cells. How does it work?
Yeah, and we think that's another significant benefit to our technology. The fact that we do not genetically modify your cells eliminates a significant risk factor that has been seen again, I keep referring to CAR T, which is clearly the leader in the cell therapy space, where there is genetic modification and where the FDA recently put black box warnings on CAR T therapy because of the potential to cause other secondary cancers. So we don't have that. The way PhagoAct works, and this is essentially the IP that we have licensed from the inventors' laboratories at Georgia State University. PhagoAct is a combination of cytokines, oligonucleotides, and other materials that when the patient's macrophages are cultured in the laboratory in the presence of PhagoAct, we elicit what the product that we call SIRPant macrophage, and that macrophage has two significant characteristics. One, it is activated to be very aggressive in its phagocytic activity. I'd like to say we make it hungry, and it's hungry for cancer, and that's what it goes out and seeks. And two, we have downregulated a negative regulator protein that macrophages normally produce called SIRP alpha, hence the name of our company, SIRPant, which is a play on SIRP and the Greek letter alpha. And by eliminating or reducing SIRP alpha expression, it does two things. SIRP alpha is the protein that prevents the macrophage from phagocytizing cancer cells, because the cancer cells express something called CD47, or the don't eat me signal, and SIRP alpha recognizes it and says, I won't eat you. By taking away the SIRP alpha recognition, we ignore the don't eat me signal and we eat the cancer. The macrophage, the company's macrophages. And then SIRP alpha is also a control mechanism that downregulates the macrophage's ability to communicate with T cells and B cells, essentially the ability to communicate with the white cells that it needs to communicate to. So again, by reducing SIRP alpha expression, the macrophage is extremely capable, very, very capable, and has an enhanced ability to program the T cells and the B cells and the other white cells that play such an important role in the immune response.
Walk us through the process of going from a patient to treating them.
Yeah, obviously we're in clinical trials now, so it limits. The patients have to first go through a review and a qualification that allows them to come into the study. In early studies like this, the FDA, of course, requires that these patients be at a later stage in their disease, having failed all approved therapies. We don't want to risk anybody who hasn't had all the therapies that have been approved that could help them. Now, once that occurs, if they qualify, we schedule they go through a screening process. We do some blood tests. We do some scans to measure tumor volume, and then they go through what's called leukapheresis. So they go to the treating hospital, or it can be a blood center, where they're hooked up to a machine for about 3 hours. The blood's drawn out of their body, goes through the machine. The machine selectively harvests white cells from the circulating bloodstream, and then puts the remainder of the blood back into the patient. So it's kind of like you're just hooked in with two tubes, one in one arm, one in the other. Blood comes out, goes back in, a little less white cells on what's going back in. The patient shouldn't worry about that, because within a matter of hours, new white cells come out of the bone marrow and replace it in the circulating system, so there's no deleterious effects. So after that 3-hour period, that cell pack is shipped on ice overnight to our manufacturing facility. The cells are then isolated and purified in the laboratory. They are cultured, and I should mention, these are not macrophages that we harvest. We harvest an immature cell called a monocyte. Macrophages do not circulate in the bloodstream. They're tissue resident. We harvest the circulating monocytes, which are then cultured for 5 days, where they differentiate and mature into macrophages. Those macrophages are then incubated for 2 days in the presence of phagoact. At the end of that total 7-day period, the cells are harvested, washed, and then reformulated into an injectable formulation and frozen and shipped back to the hospital for administration. That is a total of 7 days of manufacture. Prior to shipping, we have to undergo quality control testing. And it takes right now the vein-to-vein time, so from leukapheresis to re-injection back to the patient, about 14 days. We're working on shortening that to 9 days through rapid release testing methodologies being qualified now. But right now it's a 14-day process. The patient, and this is a major advantage over T-cell therapy, there's no hospitalization required. There's no pre-treatments, no onerous things like T-cells require lymphodepletion, which is a terrible treatment that even precludes some sick patients from being able to get T-cell therapy. We don't require any of that. The patient comes in, gets an injection into a single tumor in their somewhere in their body, readily accessible. And they go home. There's no hospitalization. To date, we've seen no serious adverse effects at all. The patient will get 3 injections over a 5-day period. So they come in Monday, Wednesday, and Friday, get an injection and go home. And that's the end of this therapy for them. That should be the end of therapy. Right now, we're at low doses. We're seeing some early indications of response and tumor shrinkage, elimination of some tumors in some cases, but this is at a low dose. We're currently finishing our last patient at low dose. After a 30-day observation period, we're going to triple the dose in the next cohort of patients. And there we are very anxious to see, as we expect that to be a strong and therapeutic dose, and we will know that by the end of the year.
Your lead indication is for relapsed or refractory non-Hodgkin's lymphoma. For listeners not familiar with the condition, what is it?
Again, I'll try to simplify it. It's very complicated. Non-Hodgkin's lymphoma is all lymphomas other than Hodgkin lymphoma, so it's a little bit humorous there. But NHL is actually 60 different cancers. It's any white cell blood cell cancer that is not a Hodgkin's lymphoma, and that can be 60 different cancers. We are treating or taking anyone amongst that group of cancers in our phase 1 clinical trial. It can be T-cell lymphoma. It can be B-cell lymphoma. We're taking all of those types of patients. So as I said, it is a complicated answer. There are a lot of different symptoms. You can see enlarged lymph nodes, fever, night sweats, weight loss, tiredness, bone pain, chest pain, itchiness, especially with the cutaneous T-cell lymphomas. So there's just so many different things it can be. In fact, cutaneous T-cell lymphoma, there's very few approved therapies for that. The Lymphoma Society is focused on it right now, and we have been granted orphan drug designation by the FDA for the development of this in the cutaneous T-cell lymphoma space. So it's a complicated answer. Essentially, it is white cell blood cancers of a variety of types.
Given the broad applicability of the technology, why start there?
We started with lymphoma because we already had indication that if you perturb or disrupt the axis I mentioned earlier, the SIRP alpha CD47 axis, that you can have a positive effect in lymphoma. So we went there knowing that's one of the major things that SIRPant M brings to the table, this disruption of the SIRP alpha CD47 axis, without all of these side effects associated with CD47 blockade, which was tried by other antibody companies. And the reason that has adverse effects is that CD47 is resident on many other cells, like your red blood cells. And if you blockade that, then you're killing the red cells in the body, which is not what you want to do. So we're taking a very different approach to utilizing the strength of that disruption of the don't eat me signal, but very specifically to the cancer cell. And so that was one of the reasons to lead with lymphoma. Having said that, the solid tumor space is where we see a tremendous opportunity because there are no effective cell therapies to date. And we truly believe that the SIRPant M approach holds the best promise for having an effect on solid tumor cancers.
What's known about it from studies that have been conducted to date?
Well, what we found out and what has been demonstrated is that SIRPant M is cancer agnostic, which is interesting in and of itself. We don't need to identify a predetermined marker or protein on a cancer. We've demonstrated that an activated SIRP alpha depleted macrophage will phagocytize all 60 of the NCI cancer panel. So NCI has established 60 different cancer cell lines across many, many different cancer types. And we've demonstrated the ability to phagocytize all of those. And then we've gone into animal models and demonstrated the ability to cure the animals of, and I'll just, some of them, lung cancer, lymphoma, colon cancer, pancreatic cancer, ovarian cancer. All of these animal models, we don't reduce the cancer or slow its growth. We cure the animal. And then what's really interesting and which is very promising for this therapy is you've cured them through an immune response, and our immune system has memory. That's why vaccines work for people in other diseases. What we've shown in our animals is when we cure, for example, colon cancer in the mouse, for the rest of that animal's life, we can go back and inject the colon cancer back into the mouse, and it will not establish. The animal has immunity to it. It attacks it and kills it immediately. And so we're looking forward to that potential in humans as well. In our solid tumor trial, one of the areas we're going to focus on initially is head and neck cancer. Head and neck cancer, 50% of the patients go through what's called curative surgery, and I'd say 100% of the patients go through surgery. 50% of those patients will recur within 2 years because there were microscopic metastases elsewhere that they couldn't get in the surgical procedure. And so if we can inject that tumor and then remove it a few weeks later, our hope is we will have immunity and that will then go out, seek out an attack and prevent that recurrence. So that's the kind of things we're looking at. Lung cancer, another example, you find lung cancer early, you remove the lobe, and that patient is likely going to recur within 5 years if we could prevent that. So not only do we want to do the initial intervention and cure, but our hope is that we'll also be able to significantly extend or eliminate the risk of recurrence post-cure, post-treatment.
And what's the development path forward?
Well, right now we're in the phase 1 clinical trial in NHL. We have completed the first 3 patients who were monotherapy low dose. This, of course, is defined by the FDA in our investigational new drug application. We are now, as soon as patient 3 completes the 30-day observation period for toxicity, and we've seen nothing to date in the first 2 patients and nothing in the 3rd patient to date. As soon as that patient clears 30-day DLT, we're opening up 2 additional arms. One arm is the same low dose, but in combination with low dose radiation of the single injected tumor. Why do we do that? With the low dose of radiation, it's not meant to be curative or ablative. It's meant to inflame that tumor because our macrophages love inflammation. And so if we inflame the tumor with a little bit of radiation, they're even more aggressive and more effective. So there'll be 3 patients in that arm of the study. And parallel, we will be recruiting patients for the high dose. The high dose is more than triple the low dose. And it is the dose that we predict from the animal studies to be fully therapeutic. That is our hope. And those patients will start enrolling in the next 2 months. And then finally, when the high dose 3 patients clear their observation period, which is a 30-day, it's called DLT, dose limiting toxicity observation period. If we successfully clear those 3 patients, we will then add 3 more patients at the high dose, again with the low dose radiation. So that is the lead program. And from that, we will identify responses and hopefully identify subgroups of the NHL population that we can then expand into working towards approval. And then we would like to initiate our solid tumor trial. Again, the first 3 patients at low dose will be what we call an all-comers trial. If the patient meets criteria, it will be across a variety of cancers. And then very similarly, we will go to low dose plus radiation. But we will also go to low dose plus a checkpoint blockade such as Keytruda. And then we will also be going to a high dose. And post the high dose DLT period, we'll then go to high dose plus radiation and high dose plus a checkpoint blockade. And our hope is that at the end of these 2 studies, we will see responses, complete responses, and we'll be able to lay out an approval pathway from that informative early work.
There are a large number of conditions beyond cancer that involve immune dysfunction. We're starting to see cell therapies applied to autoimmune conditions. What's the potential for SIRPant's therapeutic approach beyond cancer, and what plans, if any, do you have for pursuing these?
That's interesting as well, because within our patent portfolio that we licensed from the university, there has been work done on downregulating, which is what you do with the macrophages in an autoimmune condition. You've got hyperactive macrophages that are creating problems. And so it's the opposite of what we want to do with cancer. We want to tone down the response. And there has been work done at the university and in our patent portfolio that we've licensed. And one of the key PhDs from the university setting is now an employee of ours. And we've already published 1 paper working into that area and towards that opportunity. We've been a very frugal and a very small company intentionally in what I like to say we're built for this financing environment of today. And we So we have not gone after that at this point in time. We need to further develop the company, further finance the company, but it is clearly in our sights to work with the macrophages and down regulating their activity and trying to eliminate some of the hyperactivity found in autoimmunity and also in chronic infection.
Robert Towarnicki, CEO of SIRPant Immunotherapies. Robert, thanks so much for your time today.
Thank you so much for giving me the opportunity.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.